another video session from Bioremedial online classes. I am Santosh. I welcome you all to this video session. Students, today we will understand a new concept on genetics, incomplete dominance. Students, after Mendel's experiment, after Mendel's law, further research found there were some mistakes, there were some drawback in Mendel's principles. So one of the German scientists, German botanist, Karl Krenz, who was among the three who rediscovered Mendelism. So he found in a monohybrid cross that none of the two characters were present, were dominant in his experiment, and he found both the traits from the monohybrid cross were incompletely dominant in F1 generation. So this phenomenon is all about incomplete dominance proposed by Karl Krenz. So we will understand the entire concept of incomplete dominance, which is deviation of Mendel's principle. This incomplete dominance further led to discovery of many more deviations of Mendel's principle. We will also understand one after other what are the deviations of Mendel's principle. So now we will understand incomplete dominance. Students, in incomplete dominance, as the name suggests, that none of the characters completely dominant in F1 generation. So we can define incomplete dominance as the phenomenon in which none of the two characters completely dominant. That means both the character appears but not completely, is called incomplete dominance. Students, here both the characters appears partially, merely they are present in one organisms. So what did the Karl Krenz do in his experiment? He selected a 4 o'clock plant. So in 4 o'clock plant, the scientific name is Mirabilis jalapa. So he crossed a plant with red flower and a plant with white flower. When he crossed a plant with red flower, and plant with white flower. So what did he found? The red flower is denoted by 2 capital R and this is 2 small r. So just like Mendel's monohybrid cross, he crossed these two plants, red flower and white flower, and he got the gametes capital R, and he found the heterozygous condition in F1 generation. In F1 generation, he found the genotype is capital R, small r, but the phenotype is all are pink. So instead of red or white, he found a new color, that is pink. But pink is a combination of both red and white. This indicates that in the F1 generation, both red appears, white appears, but none of them completely appears or completely dominant. This phenomenon is all about incomplete dominance. So Krenz, when he found in F2 generation, what did he got the result in F2 generation? In F2 generation, he found 2 types of gamete here, capital R and small r. So similarly, when he self-crossed or self-pollinated the F1 generation pink plants, he found if you cross the 2 varieties of gametes, male gametes and female gametes, he found here 2 capital R, 1 capital R small r, again 1 capital R small r, 2 small r. So this is the genotype he found. But the phenotype he resolved, this capital R, just look at this capital R will give red. That means red, he found 1 plant. Then 2 capital R small r, which gives pink. So pink he found 2 plants, Out of 4 plants, he found 1 red, 2 pink, and finally smaller it represents white. So he also found 1 plant white. So the phenotype ratio, the phenotype is, we can write down 1:2:1. Let us look at the genotype. If you see the genotype, you see 1 capital R, capital R. So there is no capital R, capital R. So 1 capital R combination, capital R smaller combination you will find in 2 places. So 1:2. Then smaller combination is 1. So here genotype is also 1:2:1. So incomplete dominance is the only phenomenon where you will find both genotype and phenotype is same, 1:2:1. So this is all about the concept of incomplete dominance experimented by Karl Lorenz, a German botanist. After discovering the Mendelism, he found somewhere the Mendelism or the Mendel's laws deviated. So this is one of the deviation of Mendel's law. We will understand further deviation of Mendel's law in the next videos. Stay with me to understand, to get more and more videos. If you like this video today, please like, share and comment. Do subscribe my channel if you have not subscribed yet, and follow me in my next videos. Bye bye.